Okay, so we're here at Biologists at 100 with Matteo Allegretti. You've been presenting a poster at this conference, and in that poster you have a preprint. And I was wondering if you could tell us all about your preprint in about a minute.
Yes, yeah. So our preprint is about the nuclear envelope of sperm cells. These are very specialized cells, and we thought that when we started this investigation, that this protein that make the nuclear envelope had to be very specialized. So we know that in somatic cells you have a big protein complex called the nuclear pore complex that mediate the traffic of molecules in and out. And this is fundamental for separating transcription from translation and other fundamental cellular functions. So we looked at this in sperm cells that during the differentiation lose 90% of their nuclear volume. And when we solve the structure using cryotomography, that is, so we freeze the cell, we cut some section of the cell, and then we select different of this series of these nuclear pore complexes, and we average them. We've seen the architecture of the pore is very, very different. They lose mostly of the protein that make the pore. There are more than 1,000 proteins. They only a few hundred are left in the middle of the pore. And then we look at the function. And the function says that the diffusion of molecule in and out of the nucleus is really reduced. And we do this using fluorescent recovery after photo bleaching of small dextran molecules. And then we look also at the active transport of molecules, and this is also not happening. So our hypothesis at the moment is that why the nuclear pore is so different is because the sperm cells do not really need to have any active transport of molecule inside the nucleus. Their goal is just to deliver the DNA, so they don't want to have any perturbation of the DNA that is packed inside the nucleus and just has to travel to the egg.
Nice. That sounds like a really cool preprint. I mean, I was wondering, in that whole story, what did you find to be the most challenging aspect of getting all of that work to being a preprint? What was the biggest hurdle you had to overcome?
Yeah, at the beginning we solved the structure. When you have the structure, the main problem was to get some functional data and to try to put it in the context of the development. That was the hardest part. And to do that, we had to get some ethics approved. So to get some ethics approved was difficult. And to deliver also molecules like dextran inside the sperm was also very hard because it was not a canonical human cell, it was very fragile. So we struggled to do that at the beginning, then we found a good protocol.
The cell kept breaking?
It was electroporation,
Oh, yeah.
but yet it's different from like classical skin cells, so it took some time to have that running. I think this to get some ethical approval, to get tissues in, where we could also testing antibodies. Antibodies in tissue at the beginning was hard to get them in, so we had to optimize that kind of stuff. Yeah.
Nice. Okay. And obviously we're at the conference and people are on their way to the next session. But I was wondering, in your poster session yesterday, what was the most useful feedback you got from people related to this preprint?
Yeah, I think the people are interested mainly in this conference is how this nuclear pore gets so reduced during development. That's also what the reviewer want to know. So partly what the people want to know and what the reviewer want to know is how you can drastically reduce a so big protein complex to a reduced form during the differentiation or spermatogenesis in this case. So what we are trying to address this is trying to isolate the different cells during development and trying to see which kind of post-translational modification you have at different proteins, because we think that this is the key to actually not have them assembled back in the big machinery.
Yeah. And so you mentioned you're doing the revision, so the future is obviously you get on your way to a publication. But how do you think the experience of having a preprint has benefited you personally in maybe in your career, or just in general for you personally? How was the preprint beneficial?
I mean, for us, being able to put the things out, it's clearly a lot of release of stress.
Yeah.
So this is good for the lab in general. For, people have good mental health if they put the things on.
Yeah.
They don't have to wait. But in general, it's great because we have quick feedback. We can put it on our CV. So if you apply for a conference or if you apply for grant, like my postdoc is doing, is great. You have a bioarchive so everyone can see. And this is also great for the public because, you know, many of the journals are not open access. Instead, you can just look at the preprint. So it's great for general public, even if the general public probably will not understand what is written there.
Yeah.
But it's still great to have it online free.
Yeah, I mean, so my final question was, what do you think the importance of preprints are in the publishing system? But I think you've very much just answered that one. So unless you have anything else to add,
Yeah, it's really great for us is to have everything, get quick feedback, get it out, and being able to put it on our CV and present it is the big advantage.
Yeah.
Very fast.
Nice. Okay, so in that case, I think thank you very much for agreeing to do this interview, and we'll put the links to the preprint at least in the comments, and hopefully we'll see it in a fully published peer-reviewed version soon.
Thank you for asking me to do this and for being here at the Company of Biologists.